Welcome to Life in Biology. I'm Dr. Joel Graff. This video is going to be about transcription regulation. We're going to start off talking about transcription factors, and then we're going to tie it into using the CRISPR-Cas9 system to do the gene regulation for us. So without further ado, let's jump into regulating transcription. We've got the central dogma of biology here. We've got DNA going to RNA going to protein. That's the flow of the genetic information. And I have two arrows for replication, for transcription, and for translation. The arrows are of different sizes, and the point of that is that the rate at which these different steps happens can be regulated. We've already talked about regulation of the DNA replication, those checkpoints within the cell cycle, that sort of thing. So now we're moving on to regulation of transcription rates. So transcription is controlled to a large part by transcription factors, and there's two flavors of transcription factors. Some transcription factors can be activators, and that would speed up the rate at which you go from DNA to RNA. And some transcription factors are repressors, and those can either block or slow down the rate of transcription. So how that works with a regular transcription factor is that there's two bits to a regular transcription factor. So here I've drawn a transcription factor as two ovals. Usually when I draw a protein, I just draw it as one. But the point that I'm trying to drive home is that there are two separate domains within this protein that fold separately, and they each have a different job to do. Down here where it says BD, this is the part of the protein that folds up, and it has specific ability to bind to DNA. So we're going to call that the binding domain. Now it's connected, so it's the same protein, but it's connected to this other domain, which we labeled AD, which in this case stands for activation domain. And when you have a transcription factor that has an activation domain, bind to a recognition sequence this is the promoter region, the region just upstream, which is just 5' of this transition start site. That is where your transcription factor binds, and the binding domain has a specific sequence of DNA that it wants to stick to. So that's how you get this brought into a correct part. There's lots of different transcription factors in a cell, and this one has a specific recognition for whatever this sequence is. In the last video, I made a big deal out of drawing double-stranded DNA all the time. In this video, this brown line here represents double-stranded DNA. This box also represents double-stranded DNA, but I drew the box on top of the line to indicate this is where a particular gene is located within the genome. So this is genomic DNA sequence. We get the transcription factor binding, and then it pulls in. I've written pull 2 here. So there's different types of polymerases. This one, pull 2, is the one that in human cells at least is the one that would make your mRNA. And so this transcription factor's job is to recruit in this polymerase, and there's different ways that it can recruit in, and I'm not going to get into that in this video, but that would pull the polymerase in. The polymerase starts working its way down the genome, and it gets to this part that I have indicated with this arrow. That's the transcription start site. Down here, I didn't label it, but there's a transcription stop site. So it'll start making the RNA as it's moving through this gene, and then at some point it lets go of the DNA. And then it could get recruited to make another strand. So anyway, that's how a normal transcription factor works. This particular one has an activation domain, but other transcription factors can be repressors. I got my cat doing some meowing. My cat's name's Charlie the Ore Digger. That's the mascot here at Montana Tech, the school that I teach at. Anyway, moving on, let's talk about CRISPR. I have a whole series of videos on CRISPR. I'll link to several of those videos in the bottom, in the show notes. Until then, now my dog's wanting out. So this is a great video. So we've got dead CRISPR activation or not. So let's walk through this. I've got two versions of the genome where gene X is attached, or gene X is shown. So I'm basically drawing the same part of the genome as I had in the last slide. The difference here is we don't have a transcription factor per se, but instead we're going to be controlling gene expression with Cas9. That's the enzyme in the CRISPR system that most people use. And Cas9 is a protein, and it uses a guide RNA, that's what the gRNA stands for, to find a specific sequence to bind to. So in the previous slide, the transcription factor activation or binding domain was a protein that had specific recognition for a specific DNA sequence. Here, the Cas9 protein, it does not have specific DNA binding properties until it's in a complex with the guide RNA. So the guide RNA helps Cas9 figure out where on the genome to bind, and then we have a lowercase d in front of this Cas9. Cas9 is usually an enzyme that's capable of cutting DNA, double-stranded breaks, but there's two nuclease domains in there. Each nuclease domain is in charge of cutting one strand of DNA, and in this particular version of Cas9, both nucleases have been mutated to where they don't work. So instead of having the DNA get cut when Cas9 binds to a particular spot that its guide RNA draws it to, in this case, the Cas9 will just sit there and bind and just hang out. Because we've got this Cas9 expressed as what's called a fusion protein, we've included an activation domain, and that will pull in the polymerase II, and you'll get transcription of gene X, and you get the mRNA being made. So if you take this dead Cas9, connect it to, or have it guided by a guide RNA, and you have an activation domain connected to it, you can directly control and express whatever gene you want as long as you have the guide RNA bind to its promoter region. To contrast that, we have here and this is called CRISPR-A for activation. This is going to be CRISPR-I. I meant to write that on here, but CRISPR lowercase I for CRISPR inhibition. And again, we've got a guide RNA. It's causing this Cas9 to bind to the same site of DNA. However, instead of being fused to an activation domain, this Cas9 is fused to a suppressor domain. I wrote CRAB because that's just an example of one of the repressors. It's Kruppel factor. But anyway, this is a repressor, and so it would not bring a polymerase in. Instead, what it does is it causes the DNA to condense a bit, so it even lessens the chance that a polymerase would get recruited. And so then you don't get transcription and you don't get the mRNA being made. So with CRISPR-A, you use an activation domain, it can turn on a gene. With CRISPR-I, we have a suppressive domain, and it'll cause the gene expression to be turned off. And that's just that's a great example of biotechnology, where you take something from the biological world, you mix and match the pieces, and you end up with a tool that allows you to control gene expression. So you have custom transcription factors. All you got to swap out the guide RNA, and you can control any gene you want. All right, good talking to you. Have a great day. Like and subscribe, or not.